In this video, we'll be talking about one of the major functions of the reproductive system. It's really the main function of the reproductive system in males. This is the production of the gametes. The gametes are basically the sex cells, the cells that come together during fertilization to form a new life. Sperm cells in men and the egg or ovum in women. In women, of course, the reproductive system has a second function, and that's the nurturing of developing offspring. But both men and women need to form gametes. Before we get into how gametes are formed, we need to talk a little bit about how humans reproduce. Humans reproduce sexually. We have one male and one female coming together. Barring futuristic science that may allow males and females to reproduce independently, this is how the human species does it. What this means is that genetically we are a combination of our two parents, and I've tried to illustrate that here by showing the orange mother and the blue father coming together in the mix, the combination of their offspring. Humans have two sets of 23 chromosomes, one set from mom and one set from dad. So that's a total of 46 chromosomes total. These paired chromosomes are homologous to each other. That just means that they carry information for the same characteristics, like eye color or freckles, but each may carry different versions of that gene. So, on the chromosome you get from your mom and the chromosome you get from your dad, there may be a spot on that chromosome that codes for eye color, but you might have a gene specific for, say, blue eyes from your mom and a gene specific for brown eyes from your dad. So they carry information for the same characteristics, even though that information may come in slightly different flavors. What you're looking at here is called a karyotype. It's an image of an individual's chromosomes. Well, actually, this is a computer simulation of an image. It's not an actual photograph. But I'm showing this because it illustrates the fact that we have 23 pairs of chromosomes very nicely. When I say that the chromosomes come in matched pairs, I mean that they have, apart from having the similar information, or information for the same characteristics, which you can't really determine unless you sequence the chromosome, you can line them up and figure out which are paired by looking at size, so, length of the chromosome, and by also looking at the banding pattern. See how the banding patterns for the two chromosomes 1 is the same, and for the two chromosome 2s, the banding pattern is identical. So you have 23 pairs, 1 through 9, 10 through 18, 19 through 22. Chromosomes 1 through 22 are referred to as the autosomes. And chromosome 23, that 23rd pair, are your sex chromosomes. These are the chromosomes that go a long way towards determining biological sex. Males will have one X chromosome and one shortened Y chromosome. People who are biologically male. People who are biologically female have two X chromosomes. What this means is that it's actually the dad that determines the gender of the child, because the mom can only contribute an X chromosome. That's all she has. It's the sperm cell that actually determines whether the child receives a second X chromosome or a second Y chromosome. Of course, gender can get a bit more complicated. You can have people who are XXY or people who are XO, meaning that they only have one X chromosome. You can also have people who are chromosomally, chromosomally male, but are unresponsive to testosterone, and so they never develop male sexual characteristics. Gender can be quite a bit more complicated than just your chromosomes. I want to introduce a little bit of terminology here that will help you as you are reviewing the process of gamete formation. The first is the term diploid. Humans are referred to as diploid organisms because the vast majority of cells in our body, the somatic cells, have two sets of chromosomes. Di means two. So the diploid number is the number of chromosomes that we have with two full sets. Somatic cell is just the opposite of gamete. So somatic cells are basically every cell in your body other than the gametes. And so in humans, the diploid number is 46. And sometimes that 2n is used just to indicate that it's two sets. All right? Now, the opposite of diploid is haploid. Haploid is the number of chromosomes found in one set. So the haploid number for humans is 23. Now, this exact number varies from organism to organism. For example, dogs have 78 chromosomes. 78 is their diploid number. That means that their haploid number is actually 39. Cats have a diploid number of 38. So cats have two complete sets of 19 chromosomes. And 19 is their haploid number. The haploid number is half of the diploid number. It is also the number of chromosomes found in our gametes. So our somatic cells are diploid. Our gametes are haploid. So an egg cell only has 23 chromosomes in humans, and a sperm cell only contains 23 chromosomes. One complete set. So chromosome 1, chromosome 2, one copy of chromosome 3, one copy of chromosome 4, on, etc., until one copy of the sex chromosome. X chromosome in women, either X or Y in men. It's good that our gametes are haploid. This way, when the egg cell and the sperm cell join back together during fertilization, you have a diploid organism. You have 23 chromosomes joining together with 23 chromosomes to make an adult with 46 chromosomes. If the gamete cells weren't haploid, if you had an egg cell and a sperm cell with 46 chromosomes, the offspring would have 92 chromosomes. And then they'd make gametes with 92 chromosomes apiece, and their children would have 184 chromosomes. And it would just keep getting worse and worse and worse with each generation, and it would get messier and messier, and there would be more and more mistakes. The species would not survive more than a few generations. So, it's vital that the number of chromosomes in the gametes be half that of the number of chromosomes in the somatic cells. So, and how do we get to that haploid number? Through a process of cell division called meiosis. Meiosis is a very specialized cell division. It is used only to make the gametes. That is the only situation in which your cells undergo meiosis. And that goes, meiosis takes a cell with 46 chromosomes and ends with daughter cells with only 23 chromosomes. The more common form of cell division is mitosis, and mitosis is the form of cell division that's responsible for just about everything else that your cells do. Replacing damaged tissue, just replacing old tissue, making blood cells, making new bone and muscle cells as you grow. That's all mitosis. Meiosis has only the one function, and that is the topic of this video. You should already be familiar with the process of mitosis. I'm not going to go over that in detail in this video. What I will go over is meiosis, and I'll talk about how meiosis compares to mitosis. In mitosis, you start out with a cell with 46 chromosomes, although I've only illustrated 4 here. So 46 chromosomes, 2 sets of 23, and in each daughter cell, you have an identical number of chromosomes. Each daughter cell has the same 2 sets of 23 chromosomes. Now, in meiosis, you start out with a cell that has 46 chromosomes, but it undergoes 2 rounds of cell division. You have meiosis 1, and you have meiosis 2. And by the time you're done with meiosis 2, you have 4 daughter cells with 23 chromosomes apiece. 1 set of 23 chromosomes. So it's not just any random 23 chromosomes, it's 1 chromosome 1, 1 chromosome 2, 1 chromosome 3, 1 chromosome 4, 1 chromosome 5, etc., etc., etc. Actually, the chromosome number is halved by the end of meiosis 1. But we'll talk a little bit about the details of how that happens. Before we get into that, I do want to go over some terminology with you, and it includes defining what exactly is a chromosome. So a chromosome it's a long DNA molecule. When it's packaged up in the nucleus, it's wrapped around its histones. It's all packaged tightly into the nucleus. But for our purposes, we can just think of it as a big, long DNA molecule that's, carefully packaged. Now, when a cell undergoes DNA replication, which it has to happen, this has to happen before that cell divides. DNA replication happens before a cell goes into mitosis. It also happens before a cell goes into meiosis. After a cell has made a copy of its DNA, you have a chromosome that has that now consists of 2 identical copies that are stuck together, and they're stuck together at a protein complex called the centromere. Now, these 2 identical copies are referred to as sister chromatids, and as long as they are physically stuck together, they're still considered 1 chromosome. So before DNA replication, 1 chromosome is a single chromatid. After DNA replication, 1 chromosome is 2 sister chromatids joined together at the centromere. Let's just take 1 more minute and show what that would look like, incorporating our understanding of humans being a diploid organism and having 1 copy of chromosome 1 from the mom and 1 copy of chromosome 1 from the dad. So if we look at a cell before DNA replication, I'm not going to sketch out all 46 chromosomes because that would just take a ridiculous amount of time. But I'll show, say, chromosome 1 and chromosome 2, or chromosome long and chromosome short. Now you have 1 copy of the long chromosome from your mom and 1 copy of the short chromosome from your mom. You also have 1 copy of the long chromosome from your dad and 1 copy of the short chromosome from your dad. This is all before DNA replication. Now, after DNA replication, you still have 1 copy of the long chromosome from your mom, But now that one copy of the long chromosome from your mom is made up of 2 sister chromatids joined at the centromere. Okay? And you have one copy of the short chromosome from your mom made of 2 identical sister chromatids joined at the centromere. You still have one copy of the long chromosome from your dad made up of 2 sister chromatids. Yes, they're still called sister chromatids. That's just conventional terminology. You also have still one copy of the short chromosome from your dad, but that one chromosome is now made up of 2 sister chromatids. I'm stressing this terminology in the distinction between a chromosome before and after DNA replication and the distinction between a chromosome and the sister chromatids because it's important in order to follow what's happening during meiosis and why meiosis is different from mitosis and why meiosis enables you to go from a cell with 46 chromosomes to a cell with 23 chromosomes. So let's walk through that. Okay, so I've already shown you how the chromosomes differ before and after DNA replication. So now, when we go into meiosis, we start with a cell that has already replicated its DNA. So I'm going to go ahead and start there, a cell that's replicated its DNA. So now we have a cell with 2 long chromosomes and 2 short chromosomes. Now, if you remember the 4 phases of cell division, you have prophase, metaphase, anaphase, and telophase. PMAT. Prophase is the initial starting phase. That's when the cell is kind of gearing up, getting ready to divide. Metaphase is when the chromosomes in that cell are lined up in the center of the cell. Anaphase is when those chromosomes start to split apart. And telophase is at the end of cell division, as the nucleus starts rebuilding and as the cells actually separate, the beginning of cytokinesis. Meiosis 1 and Meiosis 2 go through these same phases. So you have prophase 1, metaphase 1, anaphase 1, telophase 1, and then you have prophase 2, metaphase 2, anaphase 2, and telophase 2. For convenience, I'm going to focus on what's going on in metaphase and anaphase of Meiosis 1 and Meiosis 2, because that's really where we can zero in on key differences between meiosis and mitosis, and how you get to half the number of chromosomes. So, if we take this cell and we progress on to metaphase 1, the homologous chromosomes, so the 2 long chromosomes, line up at the center of the cell. So you have the long red chromosome and the long blue chromosome kind of associating with each other. And you have the short red chromosome and the short blue chromosome associating with each other. These little associations are called tetrads because there's 4 sister chromatids. They're called tetrads. And these are kind of nifty because when these guys line up, when these guys get really close to each other, they kind of intertwine with each other. And you can have something called recombination occurring. Recombination is also known as crossing over, and that happens when you get a small piece of one chromosome exchanging DNA with a piece of the homologous chromosome. So this is why it's called the crossing over, because there's this little point there where the chromatids cross over. And so when they separate, you'll be left with chromosomes that look like that. This crossing over actually happens in prophase 1. Although since the chromosomes aren't fully separated until anaphase, really they aren't completely finished with the crossing over until then. But the association, the formation of the tetrads, the association of the homologous chromosomes, and the beginning of crossing over, that happens during prophase 1. Now, since the homologous chromosomes are paired up, and since it's the homologous chromosomes that line up during metaphase 1, when you get formation of the meiotic spindle in this case, the spindle fibers from either side of the cell are attached to different homologous chromosomes. Okay, so the spindle fibers from one side of the cell attach to your mom's chromosomes, the red chromosomes, and the spindle fibers from the other side of the cell attach to the blue chromosomes. It's not, you don't have all of the red chromosomes going to one side and all of the blue chromosomes going to the other side. I could just as easily have drawn it like this. It's random. And that's why you end up with a random assortment of your mom's genes and your dad's genes in your offspring. Recombination, the crossing over that happens during prophase 1, this also contributes to the variety, as well as the randomness of the distribution of the chromosomes. And variety, when you're looking at a species perspective, variety is a very, very good thing. If we all were genetically identical, if the entire human race were completely genetically identical, all it would take would be one super flu to wipe everyone out, and the human species would be done. But if we have a lot of genetic variety, then chances are pretty good that some people are going to survive and carry on the species. So genetic variation is really, really a good thing. Okay, so since those spindle fibers have attached to opposite homologous chromosomes, now when we get to anaphase 1, what's going to happen is that the homologous chromosomes are going to move to opposite sides of the cell. So now your mom's long chromosome is separated from your dad's long chromosome, and your mom's short chromosome is separated from your dad's short chromosome. Okay? So, when telophase happens in cytokinesis, and the cell completes its first meiotic division, the end of meiosis 1, you are going to have two daughter cells with different genetic makeups. And in one of those daughter cells, you'll have your mom's long chromosome, and in the other one you'll have your dad's long chromosome. And you'll have your dad's short chromosome in one daughter cell, and your mom's short chromosome in the other daughter cell. But now remember that terminology discussion we had. Each chromosome is made up of two sister chromatids, but it's still considered to be one chromosome. So, in humans, at the end of this first meiotic division, Now you have 23 chromosomes, but a total of 46 chromatids. So meiosis 1 is really the place where there's big differences between meiosis and mitosis. Meiosis 2 really proceeds very similarly to mitosis, with one exception, and that's that there's the difference in the number of chromosomes. So here, we're looking at the daughter cells from meiosis 1, and now let's see what happens to them in meiosis 2. So in meiosis 2, in metaphase 2, you have the chromosomes lining up in the middle of the cell, just like they do in mitosis. You don't have homologous chromosomes lining up anymore, you don't have tetrads forming anymore, because there is no homologous chromosome left in these cells. You just have one long chromosome and one short chromosome in each cell. Now, in Anaphase 2, this is one difference. In Anaphase 2, that's the point at which the sister chromatids split. And so basically the centromere is breaking down. The centromere that joins those sister chromatids together, it breaks down. The sister chromatids are pulled apart. Remember, in Anaphase 1, the sister chromatids are still joined together. The centromere is still intact. At this point, Anaphase 2, now you have one long sister chromatid drawn to one side of the cell, and one long sister chromatid drawn to the other side of the cell. One short sister chromatid drawn to each side of the cell. And same over here in the other cell. So now, when telophase and cytokinesis happens, now each daughter cell has one long chromosome and one short chromosome. So if we're bringing it back to the number of chromosomes that humans have, each daughter cell now has 23 chromosomes, each one made up of a single chromatid, so 23 chromatids at the end of meiosis 2. This chart summarizes the differences between mitosis and meiosis visually. So it's pointing out some of the key differences between metaphase of mitosis and metaphase 1 of meiosis. In mitosis and metaphase, you have individual chromosomes aligning at the metaphase plate. In meiosis 1, you have tetrads aligning at the metaphase plate. And look, you can even see the crossing over, the recombination happening right there. In anaphase of mitosis, you have the sister chromatids separating. But in anaphase 1 of meiosis, the sister chromatids are still intact. Each chromosome is still made up of two sister chromatids. So you have the homologous chromosomes separating. And the sister chromatids don't separate until you get to meiosis 2, anaphase 2 of meiosis 2. The last slide was a visual illustration of the differences between mitosis and meiosis. This slide is just a verbal clarification, just laying out the differences between mitosis and meiosis 1. And I just want to illustrate one more time. Here you have the sister chromatids separating in anaphase of mitosis. In meiosis, this does not happen until you get to Anaphase 2. That second round of cell division that's part of meiosis. And that's how you go from the diploid number, 46 chromosomes, to the haploid number, 23 chromosomes, that make a functional gamete. Okay, after studying this video, you should be able to talk a bit about these questions. You should be able to talk a bit about the purposes of the reproductive system. Remember, gamete formation, very critical in both men and women. But don't forget about the nurturing of the offspring that happens in women. You should also be able to talk about what the gametes are, distinguish between the diploid and haploid number of chromosomes, describe the process of meiosis, and compare it to mitosis. And as part of describing the process of meiosis, you should be able to answer these questions. So at what point does the number of chromosomes reduce by half? And what is recombination? And when does that occur?